So, hi everyone, and welcome to this session on PFAS toxicology and implications for human health. This is a session in which we'll hear from several different experts on a pretty wide variety of topics related to toxicology of PFAS, as well as what that implies for further action. So first, I'd like to introduce the chair, Dr. Angela Slitt. Dr. Slitt is a professor in the Department of Biomedical Sciences in the College of Pharmacy at the University of Rhode Island. She has 20 years experience as a toxicologist, specializing in mechanisms of liver injury and biliary excretion and transport, with numerous publications in the area of toxicology. Dr. Slitt is a passionate educator as well, who's involved in toxicology teaching at the undergraduate level, and she's excited to be part of the STEEP team. So, Dr. Slitt, please take it away.
Thank you, Jared. So I'd like to introduce you to this afternoon's session, PFAS toxicology and implications for human health. Again, my name is Angela Slitt. I'm a professor of biomedical and pharmaceutical sciences in the College of Pharmacy at the University of Rhode Island, and also a project leader for the STEEP Superfund Center. So welcome back. Thank you. So I wanted to introduce this session with just kind of some perspective about the challenges with PFAS. So for the challenges for a toxicologist, in part is that there is such a growing number of PFAS, and there are so many to tackle. Because of this, it's virtually impossible for us to assess them one by one, like we've done for PFOS and PFOA. Anybody who's reviewed those very large documents for PFOS and PFOA see the body of work that it's taken just for two. And so the question is, how can we do this for such a large number of new chemical entities? And then another question that toxicologists face or ask is whether or not the newer PFAS, emerging PFAS, replacement PFAS, behave in the body, in the cell, like legacy PFAS. Do they have similar toxicodynamic patterns? Do they have different or similar toxicokinetics compared to the legacies? Will they be short half-life? Will they be a long half-life? Will they be bioaccumulative? And then what tools can we use that we feel can improve the predictions to handle these thousands of PFAS in a timely way? And can we work together to make safer replacements? So there's gaps and needs in PFAS tools. Some of the tools we have at our hands are really good, whereas others there's gaps, and others may not completely recapitulate the effects that we see in humans. So with regard to rodent models, there's obvious differences in nuclear receptor signaling pathways, such as the PPAR alpha pathway, and that always is a bit of a confounder when we're going from rodent to human response. A good thing about rodent models is that they can express various systems for PFAS absorption and distribution and excretion, such as transporter systems, FABPs, CD36. They have albumin, they have fatty acids. So they have a lot of a milieu of mechanisms that help us with mechanistic insight. Sometimes, though, they're not very practical to assess thousands of PFAS for toxicity in ADME. And even still, if we used all of those rats or mice, they're still limited in their ability to help us predict in vitro, in vivo extrapolation. So there's gaps. With regard to in vitro and cell-based tools, the great thing is that we can use them in a high throughput way, and they can accommodate very large chemical libraries. And for talk 4, Eamon L. Catton will kind of bring an industry perspective to this problem. The nice thing is that they adhere to the 3 R's for reducing animals in research. They can be integrated with rodent studies to improve in vitro and vivo extrapolation, and they can also be used to study pathways very well. So they're great for assessing a toxicodynamic/pharmacodynamic response to a chemical, but sometimes they don't really help us predict the potency. And that's because sometimes there's differences in whether or not you include albumin or fatty acids in your media. Do those cell lines express transporters? Do they express FABPs? Do they really have the mechanisms that you would need to put holistically the adverse event together? So really, when I think about this, there are a lot of unknowns with how cells and tissues handle PFAS, and this was kind of one of the overarching parts of my thinking when I put together this session. And really, there's so many unknowns. We know that albumin is important for binding PFAS, and yet we still have a lot of questions about how albumin dictates elimination and tissue binding. Within the cells, we know that going from the blood to the cell or the cell to the excreta, we know that permeability is going to be a factor, and there's a need for really good, reproducible, rigorous permeability assays. And then there are transport mechanisms that bring PFAS potentially into the cell. And then once the PFAS get into the cell, the question is, who does it interact with? How important are FABPs? How important are other proteins? And then this, of course, the signaling pathways that are involved, like PPAR alpha and nuclear receptors. And then lastly, how do they get out of cells? Do they use permeability? Do they use efflux transporters? So there's so many questions. And really the questions that I have are, and what I hope we can leave this session with, are what are the relevant assays to determine these? What are the appropriate assays? Are they in vivo? Are they in vitro? And what do they look like? And I'll just give you an example from my own lab that we find kind of very interesting. Even though being a transporter group, we are interested in FABPs and protein binding in our project. And so we had, through a collaboration, gotten mice that lacked fatty acid binding protein. So they either lacked it specifically in their liver or specifically in their intestines. And we gave them a single dose of PFAS and then looked at the liver concentrations. We did look at other compartments as well, but just for today, we looked at liver concentration after 2 half-lives. And what we saw was that there was actually no significant difference in the concentration of PFAS that we saw in the liver in mice that lacked FABP in the liver or in the intestine. And so this really kind of got me thinking about what are the mechanisms that we hold as being really critical for PFAS, what ones do we need to incorporate, and how can we also design our experiments to completely address those. So the goals of the session today, first, are to learn about tools that are currently being used to assess PFAS toxicities in vivo and in vitro, and you'll be hearing from Dr. Schlesinger and Dr. Paul Friedman. And then we'll learn about some novel ADME with regard to PFAS mixtures from Dr. McDonough. And then lastly, and I think this is kind of exciting, is that we're going to learn about how other disciplines use their expertise for kind of the same challenge for PFAS. And so Dr. El-Katten will talk to us about strategies that are used in big pharma to tackle predicting ADME in humans. And I hope the goal overall for this session is really for us to come together and to decide and discuss relevant in vitro assays, in vivo assays, and think about a framework that can predict PFAS toxicokinetics and toxicodynamics and be used to create safer alternatives. So with that, we have some of our speakers. I just want to briefly introduce them today. We have Dr. Jennifer Schlesinger. She's an associate professor of environmental health at Boston University, and she's our keynote speaker. She's going to talk to us today about her really interesting findings with use of humanized PPAR alpha mice and lipid changes. And then we'll go on, and Dr. Paul Friedman is a toxicologist for computational toxicology and exposure at the EPA, and she'll be talking to us about the ToxCast assays that are being conducted at the EPA. And then we have Dr. Carrie McDonough from Stony Brook University, and Carrie's a gifted analytical chemist, and she'll be talking to us about her findings with regard to AFFF and ADME for PFAS mixes. So with that, thank you for this session. I hope you have a great session. And first, I'd like to introduce Dr. Jennifer Schlesinger. Jennifer is an associate professor in the School of Public Health at Boston University. Jennifer's laboratory studies nuclear receptor signaling, adipocyte differentiation, bone formation. Jennifer is an amazing scientist. She is a pioneer in this area for the Society of Toxicology. She has spent years gaining tremendous expertise in vitro assays to study the process of stem cells becoming adipocytes. She is an expert in PPAR gamma signaling, and she's using these in vitro assays along with mouse models to disentangle some of the very important observations that we see with PFAS, including changes in serum cholesterol levels. So with that, Jennifer, we're really happy that you're here. Jennifer's a good friend, and I'm just really excited to have her today, so thank you.
Awesome. Thank you so much, Angie. Okay, so I believe I am ready to move to my talk. All right, so first of all, I want to say thank you so much, Angie, for the invitation to share our work today. And I'm going to get right into it because I have a lot to tell you about. So recent toxicological review of PFAS by ATSDR revealed that there is ample epidemiological evidence showing that PFAS are associated with multiple adverse human health effects. Effects from increases in serum cholesterol to impaired vaccine responses to adverse birth outcomes and cancer. This is an overview of my talk today, and in my group, we investigate mechanisms connecting PFAS exposure to dyslipidemia, including increased serum cholesterol. So over the next 30 minutes, I'm going to begin by discussing the significance of the outcome of increased serum cholesterol that's been observed in the epidemiological data. I'll show you what we have done with regards to identifying and developing human relevant models to study the associations between PFAS, serum cholesterol, and serum triglycerides. I'm going to show you how we think about PFAS both singly and in mixtures. And lastly, I'm going to share some data that we've generated that are informing how we think PFOA is disrupting cholesterol and triglyceride homeostasis in the liver and the serum. So, as I said, human PFAS exposure is strongly associated with increases in serum cholesterol. Why do we care? We care because atherogenic dyslipidemia, that's defined by elevated triglycerides and decreased high-density lipoprotein cholesterol, as well as increased blood LDL, or low-density lipoprotein cholesterol, are major contributors to cardiovascular disease, which is the leading cause of mortality in the United States. So in case you don't think about cholesterol all that often, cholesterol and triglycerides are molecules that are not water soluble, so they need to be chaperoned in the blood. In the liver, cholesterol is packaged with triglycerides in phospholipid-coated particles that also contain chaperone proteins called lipoproteins. The liver then releases these particles, which are called very low-density lipoprotein cholesterol, or VLDL. And what you see in, let me pull up my pointer, hopefully. What you see here in this VLDL, the triglycerides are the yellow dots and the cholesterol are the blue dots. And you see that they're surrounded by a phospholipid layer with these chaperone proteins incorporated. So these VLDL particles that have been released by the liver are cruising through the circulation, and they are releasing triglycerides for use by tissues as building blocks or energy sources or for storage. As this triglyceride is being delivered to the tissues, results is a change in the density of the particle, so that the VLDL has now become LDL, or low-density lipoprotein cholesterol particles. And so in the process of losing all that triglyceride, the density increases significantly. So they go from very low VLDL to LDL. And what the LDL is doing now, these LDL particles, is delivering cholesterol to the cells. So not all of the cholesterol in circulation is actually needed. And therefore, there is a third major cholesterol particle type. Oops, I went the wrong direction. It is called high-density lipoprotein cholesterol, or HDL. And the job of HDL is to return unused or excess cholesterol to the liver. And this is facilitated, you'll see, by a different set of lipoproteins that are chaperoning this particle. So I want to make the point that cholesterol is absolutely necessary for life. It is the fodder for steroid hormones, and cholesterol is an important component of membranes. However, you may have heard of LDL being referred to as bad cholesterol. That's because the majority of cholesterol is circulating in the form of these LDL particles, and it's this cholesterol that can actually stick to vessels and cause atherosclerotic plaques. You also have likely heard of HDL referred to as good cholesterol. And the reason why it's called good cholesterol is its job is to get cholesterol out of the blood back to the liver so that it can be disposed of. So you can measure total cholesterol and triglycerides in blood, but that's only going to tell you total amounts. It doesn't tell you how the triglycerides and cholesterol are distributed across these different particle types. So how then do we examine the distribution of triglycerides and cholesterol across the different cholesterol particles? We collect the blood, and then we separate the particles based on their density and measure the triglycerides or cholesterol in each of the fractions. And so what you see here is a typical triglyceride profile from human serum. And so you see the vast majority of triglyceride is associated with those VLDL particles, with some being associated with LDL particles. What about the cholesterol? In human, again, this is the distribution of cholesterol across the different particle types in human serum. And you see that in humans, most cholesterol is found in these LDL particles. One of our issues in studying changes in blood lipid homeostasis is that there are different distributions of triglycerides and cholesterol across particles between humans and rodents. So let's take a look at the differences. So in the solid red line, you'll see that the distribution of triglycerides across the different particle types in the mouse, and you see that the vast majority, again, of triglycerides is found in the VLDL particles, with much less being found in the LDL particles. The cholesterol profiles look even more different, where now you see that the vast majority of cholesterol in the mouse is carried in HDL particles. This creates a challenge because chemical-induced changes in LDL in particular can be difficult to detect in the mouse model. So, speaking of mouse models, some mouse studies show that PFAS exposures result in increases in serum triglyceride and cholesterol, while others have failed to show this relationship. So a question has arisen. Do inconsistencies in results from rodent studies imply that the epidemiology is incorrect? This is an important question because some have concluded that because of these inconsistencies, the association observed in humans between PFAS and increased serum cholesterol cannot be causal, that there is not a direct association between PFAS body burden and disruption of serum lipids and cholesterol in humans. However, that conclusion fails to take into account several very important considerations. So first of all, different results have been generated in different strains of mice. Second of all, most studies have been conducted with mice fed a standard rodent diet that's low in fat and cholesterol. And this approach fails to take into account the fact that dietary lipids and cholesterol themselves play an important role in determining the concentrations of triglycerides and cholesterol in blood. Different results between increasing and decreasing serum lipid and cholesterol levels have been observed that depend upon the dose. Not all of the PFOA serum concentrations in data sets, however, are human relevant. And finally, of the 30 plus studies investigating the effect of PFOA alone on liver or lipid homeostasis in animal models that have been published since 1992 have included female mice. So I hope I've convinced you that it's critical to choose the right animal diet, the right animal, the right diet, and the right dose in your model in order to generate human relevant data to investigate not only the ability of PFAS to cause serum lipid disruption, but also to determine the mechanisms through which PFAS induce that disruption. All right, so speaking of mechanisms of action, one of my favorite topics, there's one more important thing that we need to think about in designing human relevant models in which to test the effects of PFAS. And Angie alluded to this. So remember that liver is an organ that is critical to lipid homeostasis. It expresses many genes that control multiple aspects of lipid handling, from synthesis and storage to oxidation and excretion. And it's the balance of the expression of all of these genes that will largely determine serum cholesterol and triglyceride levels. There is a nuclear receptor called PPR alpha that controls the expression of many of the genes that are involved in lipid handling. PPR alpha is best known for its regulation, its upregulation of fatty acid metabolism. So activation of PPR alpha leads to the breakdown of fatty acids for energy, to release energy. But PPR alpha plays both sides of this game and also controls the expression of genes involved in incorporation of fatty acids into triglycerides. Importantly, PFAS can bind to PPR alpha, turn it on, and change the balance across these lipid handling genes. So the role or importance of PPR alpha in humans is controversial, most especially when it comes to the role that PPR alpha may play in the effects that are induced by PFAS. So I'd like to do some myth busting. So first, it's been thought in the past that PPR alpha is not highly expressed in human liver. This is not true. Initial observations suggested that PPR alpha was not highly expressed in human liver. But the most recent studies show that PPR alpha is expressed at similar levels in human and mouse livers. Okay, next issue has been that PPR alpha activation induces liver cancer in rodents, but not in humans. This is true, but it's not relevant to the question of whether PPR alpha plays a role in PFAS-induced effects on lipid homeostasis, as the genetic programs that control lipid homeostasis and that are regulated by PPR alpha in mice and humans are largely similar. Next is that PPR alpha is a therapeutic target for treatment of serum lipids. So you give these drugs that activate PPR alpha are used to decrease, in particular, serum triglyceride levels. So it's true, PPR alpha is a therapeutic target of lipid-lowering drugs. But the relevance of this to our story about the connection between PFAS and increases in serum lipids is unclear, because we don't yet know if PFAS activate the same set of PPR alpha target genes as those highly specific drugs do. And as Angie alluded to, PFAS don't just activate PPR alpha, which I'm going to talk about a little bit later. So the last issue is that mouse PPR alpha is more efficaciously activated, is activated to a greater extent by the same amount of a ligand than is human PPR alpha. This is very much true and highly relevant to the potential effect of PFAS on liver and serum lipid and cholesterol homeostasis. So what's the takeaway? PPR alpha is relevant to humans. Therefore, using human relevant models is crucial to understanding the effect of PFAS on serum lipids and allowing us to be able to identify the mechanism or mechanisms of action. So on that note, in my lab, we are testing the hypothesis that PFAS induce dyslipidemia, and by that I mean PFAS induce an increase in serum triglycerides in the form of VLDL, as well as increase serum LDL, which are major contributors to cardiovascular disease. So which PFAS do we work with? This audience well knows that PFAS come as in the form of polymers and non-polymers. In the non-polymers, we have PFAS that fall under the category of perfluoroalkyl substances as well as polyfluoroalkyl substances. The PFAS that we currently work with are all perfluoroalkyl substances. We work with carboxylic acids, including PFOA, PFHPA, and PFNA. We also work with sulfonic acids, including PFOS and PFHXS. And then we also work with perfluoroalkyl ether acids, so newer replacements for some of these older carboxylic acids and are forming as a result of use of sulfonic acids. And these include GenX and Naphion byproduct 2. So we start with each new PFAS that we're interested in by testing its interaction with PPR alpha in a reporter assay that specifically expresses human PPR alpha. We use a synthetic, highly potent and highly specific PPR alpha ligand called GW7647 as our positive control. So let's take a look at the activation of human PPR alpha by several of the carboxylic acids that we work with. So first of all, notice that across the span of our carboxylic acids, including the newer GenX, that they are all able to maximally activate the receptor. But you'll see that PFOA and PFNA seem to be a little bit more potent than PFHPA and GenX at activating PPR alpha. Second, let's take a look at the sulfonic acids. And you see something different here. So first of all, you see that PFHXS, PFOS, and Naphion byproduct 2 all appear to have activate human PPR alpha with similar potency. But none of these sulfonic acids maximally activate human PPR alpha. So what we can conclude is that carboxylic acids fully activate human PPR alpha, meaning that they're maximally efficacious. And sulfonic acids only partially activate human PPR alpha, meaning that they're partially efficacious. So I want to take a brief detour. Most of the data that I'm going to show you today was generated in systems that were treated with single PFAS. However, that is not how humans are exposed to PFAS. We're exposed to PFAS as mixtures. And we think about mixtures in our toxicology group and how we can best model the activation of nuclear receptors, like PPR alpha, by chemical mixtures. So what you see here is an analysis of the activation of human PPR alpha by a mixture of PFAS. The data were generated in our human PPR alpha reporter system. Along the y-axis, you see percent maximal PPR alpha activation. Along the x-axis is the concentration, the total concentration of PFAS in our mixture. The black dots are empirical data. They show the activation of human PPR alpha by a mixture of 4 PFAS at ratios that they are found in human serum. We used 2 approaches to model the data. The relative potency factor approach is a modeling approach that assumes that all of the components in the mixture maximally activate the receptor. So by comparing the empirical data in the black dots and the model generated by relative potency factor approach, you can see that the relative potency factor approach overestimates the activation of human PPR alpha by our PFAS mixture. On the other hand, generalized concentration addition is a modeling approach that was developed by my collaborator, Tom Webster. And it's an approach that allows for components of the mixture to have differing abilities to activate the receptor. Not all of the components have to maximally activate the receptor. And so what you see here, so the dotted red line is the generalized concentration addition estimate of the mixture effect of on the activation of human PPR alpha by these 4 PFAS. And you see that that generalized concentration addition much more accurately models the activation of human PPR alpha by these 4 PFAS. So the take home is that PFAS mixture modeling approaches need to take into account efficacy. How well do each of the components in the mixture activate PPR alpha? Okay, so back to thinking about the carboxylic acids maximally activating human PPR alpha and the sulfonic acids only partially activating human PPR alpha. What I'm doing here is moving from our very simple but very easy, but very artificial reporter assay to cultured human liver cells. And what I want to point out, and the cultured human liver cell model that we use are differentiated HEPA-RG cells. And what you see following treatment with a carboxylic acid or a sulfonic acid in our human liver cell model is evidence for these same differences in efficacy, these same differences in the ability of carboxylic acids and sulfonic acids to fully activate human PPR alpha. In this case, we used a biomarker. We looked at the expression of ADRP as a biomarker of PPR alpha activation. This is a PPR alpha target gene. And so you see that PFOA can significantly increase the expression of ADRP in our human liver cell model, while PFAS cannot. All right, so we have these great in vitro tools to work with in studying PFAS, but in the end, the systems regulating serum lipid homeostasis are complex. We cannot test the effects of diet or sex in a dish. And further, the toxicokinetics and toxicodynamics of PFAS are also multifactorial. So we developed a new approach to test our hypothesis that PFAS are inducing serum dyslipidemia through activation of human PPR alpha. Our model includes 2 strains of mice, one mouse that expresses human PPR alpha and another mouse which doesn't express PPR alpha at all. These are both on the SV129 background, and we use both male and female mice in our exposure studies. We also use a human-based diet. So we created a What We Eat in America diet based on the What We Eat in America analysis in NHANES. Because we happen to be using young mice in these first studies, we based the ratio of the components of the diet on what 2 to 19-year-olds eat in the United States. The mice were exposed to PFOA in their drinking water, and they were exposed for 6 weeks during while they were moving from 3 to 9 weeks of age. So we measured serum PFOA concentrations in order to ground truth our observations relative to what human body burdens are. So first of all, I'll point out that we did not detect a difference in serum PFOA concentrations in male versus female mice. And given our dosing scenario, we generated serum concentrations on the order of 50 micrograms per mil of PFOA. And this is about 2 times the level of a highly exposed fluorochemical worker. So what did we observe? I'm going to start with the liver. It's not surprising that we observed that PFOA, so this is body weight, this is liver weight as a percent of body weight on the y-axis. We observed that PFOA treatment increased liver weight in both male and female mice. We also observed that PFOA treatment increased liver triglycerides in male and female mice. You can see here in the histology that lipid accumulation was occurring in microvessels, particularly in male mice. And I'll point out that female mice were actually more resistant to the PFOA-induced increase in liver triglycerides. So PFOA is causing larger livers to form, is causing livers to get bigger, and is causing triglycerides to accumulate in the liver. Are these two phenomena related? The answer is yes. You can see that in both, when we look at the correlation between triglyceride concentration in the liver versus liver weight, that there's a strong correlation between liver triglycerides in female mice, expressing human PPAR alpha, in male mice expressing human PPAR alpha. The slope of the relationship is significantly steeper in the males. So of course you're probably wondering, does this have anything to do with PPAR alpha? And we tested this in our second mouse model. And what you can see is that in female mice that do not express PPAR alpha, that there is no relationship between liver triglycerides and liver weight. In the males, you see that the relationship between liver triglycerides and increase in liver weight does remain, but the relationship has become less robust. So we've also conducted lipidomic analyses to examine the triglyceride, actually identify the triglycerides that are changing. If you're curious, this analysis is published in our recent paper. Okay, so we see that PFOA modifies liver lipid homeostasis, and I've introduced you to the fact that liver is an important regulator of serum lipids too. So now what I want to do is turn to looking at what we observed in serum of PFOA-treated mice. So here are these cholesterol distribution profiles that I introduced you to at the beginning. And I want to focus your attention on the cholesterol profiles in the male humanized mice. So what you see is, not surprisingly, the majority of cholesterol is found in these high-density lipoprotein particles in our vehicle-treated mice, with just a smidge of cholesterol found in LDL particles. When the male mice were treated with PFOA, however, we see a significant shift, a significant increase in the LDL concentration in the serum of the mice. So in the males in particular, we see not only an increase in total serum cholesterol, but we see a shift of the distribution of the cholesterol from the HDL particles to the LDL particles. So we're beginning to delve into why this could be happening, what particular pathways are being perturbed that can contribute to this change in serum cholesterol. We identified biomarker genes of 4 pathways contributing to cholesterol homeostasis in the liver, involving cholesterol synthesis, export, uptake, and disposal. And I'm sorry to say that we came to no definitive conclusions so far, but I'm going to point out a couple of things. So first of all, again, you see evidence that there is a sex difference, that PFOA is having a greater effect on expression of genes involved in cholesterol homeostasis in females than in males. And the other thing that I'm going to point out is that we see changes in multiple pathways, multiple processes that are potentially contributing to changes in cholesterol homeostasis. So you're going to have to wait for future talks when I have a clear explanation of the which pathways are changing. What's happening, so now I want to talk about what's happening with triglycerides in the serum. And this is a different story than we saw with cholesterol. So first, these are just analyses of total serum triglycerides in male mice, in either humanized mice or null mice. So if we concentrate here on the human PPR alpha expressing mice, vehicle data from concentrations from vehicle treated mice are open circles, from PFOA treated mice are black circles. And you see that in the humanized mice, that PFOA had little effect on serum triglyceride concentrations in either females or males. But something very interesting appears in the PPR alpha null mice, and particularly in the females. In the female mice that do not express PPR alpha, you see that PFOA strongly increases, significantly increases serum triglycerides. And we see this not only in total triglycerides, we also see this in our distribution analysis. So again, I'm going to have you focus specifically on the null mice. You see, first of all, that there's a significant increase in triglycerides that are associated with VLDL particles that's based on genotype alone. And that when the female null mice, PPR alpha null mice, are treated with PFOA, there is a dramatic increase in the triglycerides that are associated with VLDL particles. Okay, so again, we are starting to delve into the how and the why that this is happening. And so we've started by investigating gene expression in the liver. And as I mentioned, PPR alpha plays both sides of the fatty acid teeter-totter. It regulates expression of genes involved in fatty acid oxidation, so the breakdown of fatty acids to release energy. But PPR alpha is also regulating of expression of genes that are incorporating fatty acids into triglycerides to store energy. And what we find was that PFOA increases both the expression of genes involved in fatty acid oxidation, as well as the expression of genes involved in triglyceride synthesis. So more to come on these types of analyses and identifying pathways in future talks. I'm going to finish by returning to the question, do inconsistencies in the results from rodent studies imply that the epidemiology is wrong? And can our data and our models help to answer this question? So in order to start addressing this question, we identified two other mouse studies in the literature that used similar, although not exactly the same, experimental designs. We calculated the percent change in triglycerides relative to control mice and plotted these data against serum PFOA concentrations. I have to say that these data are only from male mice because the other studies we identified did not use female mice. The data in the hatched bar are the data from our humanized PPR alpha male mice. The red arrow indicates the level of PFOA in highly exposed fluorochemical workers. So you see that at serum concentrations of PFOA that are above the level in fluorochemical workers, you see that higher concentrations of PFOA are related to or associated with decreasing serum triglycerides. So at serum PFOA concentrations that are not human relevant, PFOA decreases serum triglycerides. If we look at concentrations lower than those experienced in fluorochemical workers, we see that triglycerides increase, serum triglycerides increase with PFOA. And then, of course, if you go low enough, at very low PFOA, serum PFOA levels, there's no effect of PFOA on triglycerides. So you're likely wondering if we see the same thing for cholesterol. The answer is yes, but the relationship isn't so tidy. And that's largely because dietary cholesterol is a very important contributor to the regulation of serum cholesterol. And there are not enough studies with similar diets or studies with multiple doses on cholesterol in mice that are eating cholesterol containing diets yet to truly define the dose response. But suffice it to say, or at least for now, we can say that PFOA is inducing a non-monotonic dose response in terms of its effect on serum triglycerides. Is there a mechanistic explanation for a dose response like this? And the answer is yes. This type of dose response simply requires that two receptors be activated by the same chemical. And lo and behold, we and others have shown that PFAS activate at least 3 nuclear receptors that regulate liver and serum cholesterol and triglycerides. We see in our human liver cell model that not only does PFOA activate human PPAR alpha, we also have new evidence that both PFOA and the sulfonic acid PFAS increase the expression of CYP3A4, which is a target gene for both PXR and CAR. We were able to better tease this apart in data from our humanized mouse model. So again, we know that in both females and males, in our humanized PPAR alpha mice, that PPAR alpha is activated by PFOA. We also see that target genes of constitutive androstenedione receptor and pregnane X receptor are also activated by PFOA. All right, so on to our conclusions. First of all, many PFAS activate human PPAR alpha, but in our analysis, only the carboxylic acids fully activate human PPAR alpha. In mice expressing human PPAR alpha, subchronic exposure to PFOA, number 1, disrupts liver lipid homeostasis in a PPAR alpha and a sex-dependent manner. In our mice expressing human PPAR alpha, PFOA exposure increases serum cholesterol and triglycerides, again in a PPAR alpha and a sex-dependent manner. We found that activation of PPAR alpha alone cannot explain the dose-dependent effects of PFOA on serum lipids. And finally, the mouse data do not contradict the human data when human-relevant doses are used and human-relevant diets are used. Okay, so before I finish up, I just want to give a shout out to 2 graduate students in our toxicology group. Emily Hamill presented a poster here on how of comparing the different definitions of PFAS. She also gave a poster talk. If you are interested to hear more about our mixture analyses of PFAS and human PPAR alpha, Graylin Nielsen will be giving a symposium talk at SOT in PFAS, Some, Science and Policy of Mixtures. And if you're curious about the sex differences that we are observing, I'm going to get a chance to talk more about sex differences also at SOT in the Let's Talk About Sex through the lenses of a toxicologist. And finally, I would like to acknowledge my collaborators at Boston University, Wendy Heiger-Bernays and Tom Webster, as well as our lipid and lipidomic expert, Tulia Hoitalainen at Örebro University. And in addition to Graylin's contribution to this work, I'd like to acknowledge the work of Catherine Boston, Jennifer Oliver, and Hannah Puckett. And with that, I would be happy to take questions. Oops. I hope to goodness I wasn't talking to myself that whole time.
Thank you, Jennifer, for a wonderful presentation. That was great. Do we have any questions from the audience for Dr. Schlesinger? Also, are there any questions through Wahova? Okay, so as we kind of see if anybody's answering from the questions. Jennifer, I have a question for you. My question for you was whether or not, I'm going to look at you through the screen this way, is to tell me a little bit more about your PPAR alpha experiments with regard to, did they include fetal bovine serum in the media? What type of cell line was used in those Porter assays?
Oh, great question, Angie. And I have to say, your work has totally turned me on to having to think about things like serum albumin and the presence of transporters. So thank you for educating me on those topics and other talks. So the reporter assays, we do not include serum albumin. We do not include serum. We did find that there is a shift, not in the efficacy, but in the potency that we calculate. So that's really an important factor. We thought about it a lot and we tested it, and also we think about it in the experiments with the human liver cell lines as well. We do reduce the serum for two reasons. Number one, binding to serum albumin, but also the potential for fatty acids in the serum itself to be influencing how PPAR alpha in particular is behaving. Uh-oh, I can't hear you now, Angie.
Let up, please.
Oh, there we go.
We'll give Jennifer one more question, and if you have additional questions for Jennifer, please put them in Wahova, and we'll save them to the end of the session.
Absolutely.
Alex, can someone pull up thank you. Okay, so one of the comments was, not sure if directly relevant to your study or not, but New Jersey's analysis of the literature concluded that for PFAS, PPAR alpha activation plays a minimal role, if any, in hepatic effects in rats. This is very different than PFOA and PFNA, for which there are both PPAR alpha dependent and independent components of hepatic toxicity in rodents. So would you like to comment on that? Can you see the question?
That is a fantastic question. And I think you've hit the nail on the head. Yes, I think that the contribution of PPAR alpha to the biological effects of carboxylic acids is very different than the potential contribution of PPAR alpha to the biological effects of the sulfonic acids. And, I wish I had all afternoon to tell you about what we're thinking about, but that difference is going to be critical in determining the balance between the other nuclear receptors as well. So that's a fantastic thought, and we're right there with you.
Okay, so thank you so much. I totally agree with you on that last point. We're going to keep the session moving. Jennifer, come join us back for the end of the session for our panel discussion. So next, I would like to introduce Dr. Katie Paul Friedman. Katie is a toxicologist at the Center for Computational Toxicology and Exposure at the Office of Research and Development at the U.S. EPA. She's currently focused on the application of new approach methodologies to understand chemical safety assessment, with additional interests in uncertainty in alternative and traditional toxicity information, endocrine bioactivity, and developmental neurotoxicity prediction and in vitro kinetics. One of her roles in the center is to run the ToxCast program. She previously worked as a regulatory toxicologist at Bayer Crop Science, with specialties in neurodevelopmental and endocrine toxicity and predictive toxicity. She's been actively involved in multi-stakeholder projects to develop adverse outcome pathways, alternative testing approaches, and the regulatory acceptance of new approach methodologies. Her laboratory background includes development of high-throughput screening assays, the combined use of in vitro and vivo approaches, including nuclear receptor reporter biochemical assays, primary hepatocyte cultures, and many other assays. She received her PhD in toxicology from the University of North Carolina at Chapel Hill. And today she'll be talking to us about the ToxCast 21 and the assays that the EPA is using for PFAS predictions. With that, Katie, we're very happy to have you today and very interested in your presentation. So I'm going to turn this over to you.
I'm Katie Paul Friedman, and I want to thank you for inviting me to share this talk with you on the subset of our targeted high-throughput screening for nuclear receptor activation, cell stress, and immunosuppressive bioactivities for a library of 147 PFAS. I joined the Center for Computational Toxicology and Exposure in the Office of Research and Development at the US EPA in 2016. My research is focused primarily on the application of new approach methodologies to chemical safety assessment. One part of my role is running what we have historically referred to as the Toxicity Forecaster or ToxCast program. Importantly, before I jump into this, I need to draw your attention to my disclaimer, which is essentially that the views expressed in these slides are my own and do not reflect the views or policies of the agency. Hello, I'm Katie Paul Friedman, and I want to thank you for inviting me to share this talk with you on the subset of our targeted high-throughput screening for nuclear receptor activation, cell stress, and immunosuppressive bioactivities for a library of 147 PFAS. I joined the Center for Computational Toxicology and Exposure in the Office of Research and Development at the US EPA in 2016. My research is focused primarily on the application of new approach methodologies to chemical safety assessment. One part of my role is running what we have historically referred to as the Toxicity Forecaster or ToxCast program. Importantly, before I jump into this, I need to draw your attention to my disclaimer, which is essentially that the views expressed in these slides are my own and do not reflect the views or policies of the agency. In this talk, I hope to give you a very high-level walkthrough of a few key areas of work for us, beginning with the chemical library of PFAS that has been developed by Grace Pedowich and colleagues, followed by the screening approach that we've taken based on our general tiered in vitro screening strategy and also described in the EPA PFAS action plan, followed by a brief overview of what we found in targeted screening for nuclear receptor activation and cell stress, which has now been published by Keith Halk and colleagues, and another piece of work on targeted screening for immunosuppressive bioactivity signatures, which is currently in preparation. And now I'd like to give you a really brief introduction to the chemical library of PFAS that we screened. The first challenge that we faced was selecting the PFAS for tiered screening, and that selection process for developing the PFAS library is described here to the left in this publication by Grace Pedowich and colleagues from 2019. Essentially, the first steps included procurability, followed by identification of the substances that would be of most interest to the EPA. There was also an effort to include PFAS with some available toxicity data already, in addition to those that were data poor and programmatically relevant. PFAS defining criteria such as greater than 3 fluorines, no aromatic rings, and a molecular weight less than 500 were used to identify PFAS among other constraints before categorizing the library into 53 structural categories, which are described within the manuscript and this figure 1. You can also see within figure 1 that efforts were made to maximize read-across potential to information that already existed, as well as to capture a broad structural diversity from this really interesting class of substances. And if you'd like, you can visit our publicly available CompTox chemicals dashboard under lists of chemicals and search for PFAS to examine our list of PFAS, their structures, and everything that is known about them, from physicochemical properties to links to other resources. Moving on, I'd like to tell you a little bit more about our tiered in vitro screening strategy and the concepts behind it. The PFAS tiered screening for bioactivity builds upon the generic CompTox blueprint designed by Rusty Thomas and colleagues here in the center, as described in this 2019 paper. The idea for bioactivity is to first collect broad Tier 1 information from high-throughput transcriptomics and cellular phenotypic profiling, and then to use Tier 2 or targeted NAMs for select biological targets, pathways, or conformation. And historically, we've referred to this part of the tiered screening as ToxCast. Of course, there's more than just hazard when it comes to screening. We need to characterize chemicals for their analytical quality control, which is particularly important for PFAS, which may have properties that make them challenging to screen. We also need to collect toxicokinetic screening data, including hepatocellular clearance, renal transport, and fraction unbound in plasma, which is important for our ability to employ reverse dosimetry in an in vitro to in vivo extrapolation of doses that would have corresponded to these in vitro bioactive concentrations. And then, as I've mentioned before, the hazard evaluation that we'd like to do, this employs several tiers, Tier 1, 2, and 3, no approach methodologies. And today, I really only have time to talk to you about some of the data that's either public or soon to be public from Tier 2 on nuclear receptor pathways, cell stress, immunosuppression, and some other bioactivity signatures. And as I mentioned before, if you're interested, you can read a bit more about EPA's PFAS action plan and the research component, which is summarized here and available online. And now I'd like to jump into some data and results. This will be an overview of information from this 2020 publication from Keith Hauk and colleagues on a multidimensional assay suite for nuclear receptor activation and cell stress. The primary results that are reported in Hauk et al. 2020 are these transcription factor activity profiling results for 147 PFAS. And the assay that we're employing is the adagene, cis, and trans factorial assays. And we have a lot of experience with this technology. We've screened almost 4,000 ToxCast chemicals, completely independent of any PFAS effort. And so we have a lot of data that we can compare to. The assays are a little bit complicated, so I wanted to spend a few minutes just telling you kind of how they work. There are two multiplex transcription factor activation bioassays that are both engineered into the HG19 subclone of the human hepatoma cell line HepG2. And this subclone is supposed to be more metabolically active than a normal HepG2. These cells were used then in these transcription factor assays. Trans assay mode includes 24 nuclear receptor reporters that were transiently transfected, and the cis assay is the complement, and it includes reporters for 45 endogenous transcription factor response elements. These transcription factors in both the cis and the trans modes regulate several stress response pathways, including oxidative stress, as well as cell growth, differentiation, and cellular metabolism pathways. Many of these transcription factors are ligand-activated nuclear receptors, including several that have previously demonstrated to be activated by PFAS, such as PPAR-alpha, ER-alpha, the protein X receptor, and the constitutive androsten receptor. So you can see now our interest in this particular assay suite because it would give us a lot of information about a lot of different transcription factors that could be involved with PFAS bioactivity. I want to spend just a minute telling you how the assay works. It's shown pictorially here in the middle of the slide. I'm going to put some words with that. Briefly, the cells are transiently transfected with the cis or trans set of reporter units and plated into 12-well plates. They're allowed a 24-hour recovery period in 1% FBS supplemented medium. and they are then incubated with the test compounds, in this case PFAS, for 24 hours. Then the total RNA is isolated and used to understand the reporter transcription units present using a quantification of fluorescence activity. Then we can understand essentially its transcription factor activity fingerprint. The chemicals were tested in duplicate and blinded format at 8 concentrations, and the data from this experiment was analyzed using the ToxCast data pipeline. The data are fold change over a solvent control, which in this case is DMSO or water, and then they're logged to transformed and curve fit, and then we can report out potencies like a 50% activity concentration. And finally, to the right-hand side of the slide, you can see this in the publication as well. There's a full listing of the cis and trans reporter elements. You can see that there are a number of groups of these, from control to cell proliferation, and include a number of nuclear receptor reporters as well. On this next slide, I've blown up for you Table 1 from Hauk et al., because I want you to have a chance to just look through some of the nuclear receptors that are included in the transfactorial assay, which is really important for overcoming some of the sensitivity limitations of the cis assay. So for example, there's really low expression of ER alpha and the pregnant X receptor in the cis assay, which is relying on the endogenous response elements. However, when we co-transfect in the full-length receptors into the trans assay mode, we now have much more sensitive assays for ER alpha and PXR. The constitutive androsten receptor and the vitamin D receptor are really not very sensitive assays at all for us. The reason for this is that for these, we're relying on the endogenous receptor expression in the host cell, and we don't have a really good trans assay version for these two receptors. And CAR in particular is tricky because it's not necessarily dependent upon a ligand to be activated. So what did we find with this technology? There are some really general takeaways that are true for the gene assay and also true for a number of the other platforms we screened, including that lower molecular weight often corresponded to more limited bioactivity. And one reason is that PFAS with lower molecular weight tended to fail analytical QC more often. And by fail, we mean the parent structure was not detected. So to the right here, you can see a series of graphs where the y-axis is the log molar AC50 and the x-axis is a code for each PFAS. And they're divided into bins of molecular weight. So starting from 148 to 228 and going up. And then this lower row of graphs starts at a molecular weight of 330 grams per mole with increasing molecular weight as we move across. You can see on the lower panel of graphs, we see a lot more chemicals with a range of potencies, and we see a lot less red. The red color here indicates an analytical QC failure to detect parent structure. Whereas up here on the upper row of graphs, we see a lot more red. Activity was not detected at all for 76 distinct structures, and over half of those failed analytical QC. And of those failed samples, we think that chemical volatilization may have played a role. Many of these have predicted vapor pressures in excess of 100 millimeters of mercury. And another thing that may play a role is the solvent we're choosing and how it interacts with the specific acid form of PFAS. So as an example, the free acid form of the chemical known as Gen X did not have a high vapor pressure, was unlikely to have volatilized, but the ammonium salt form of this chemical showed activity as a PPAR-alpha agonist when we solubilized it in water rather than DMSO. One question we get asked a lot is what the relative potency of PFAS are compared to the rest of the chemicals that we've screened in the ToxCast chemical library. So I've broken this out a little bit for Adagene to not only highlight for you some potential transcription factor groups of interest, but also to show you the relative potency of PFAS. So on this left-hand side, you can see this is all PFAS data in these box plots. The y-axis is the log 10 AC50 in micromolar units. So a unit of 1 means 10, and a unit of 2 means 100 micromolar. And you can see that almost all of the PFAS data really fell between about 10 to 100 micromolar. And then these numbers at the top of the box plots indicate for you the number of PFAS in our library that were actually active at that particular transcription factor target. So you can see the aryl hydrocarbon receptor, estrogen receptor alpha and its response element, 3 isoforms of PPAR, alpha, delta, gamma, their response element, PXR and its response element, and then RXR alpha and beta. And you can see there's a lot of activity around the PPARs. There's a lot of activity around the estrogen receptor, interestingly enough. And so we decided that was important activity to confirm with an orthogonal assay. To the right, you can see a little bit more clearly, I've added in quote unquote everything else in ToxCast versus the PFAS in teal. So now you can see we do have some chemicals in the ToxCast library that are much more potent than the median PFAS indicated by the line across the box plots. The important thing to remember here is that we're screening to identify potentially relevant targets. And so we think confirming those targets is important. So that's what we did. We employed this assay for the estrogen receptor activity confirmation called the ASEA real-time assay. This uses electrical impedance. So the more cells you have crossing an electrode, they impede the electrical signal, and that indicates cell proliferation. That was driven by the estrogen receptor in these T47D human ductal carcinoma cells. And we have a lot of experience with this assay as well. It's a component of the ToxCast estrogen receptor pathway model, if you're familiar with that model. So what did we find with that ASEA assay? And I apologize, I've turned off my video on this slide because it was obscuring some really important content behind my face. So the confirmation of this ER bioactivity was an important part of our strategy for looking at the transcription factor activity. And you can see here I've included figure 5 from the manuscript that shows some more detail about this that I'll describe. If you recall, there were 40 to 60 PFAS that demonstrated some activity at the ER alpha trans assay or the ER response element cis assay. And these assays can be viewed as orthogonal, and that reduces the set to 10. So essentially these PFAS that act at both the ER alpha trans and the estrogen response element cis indicates that we're seeing concordance, and we'd like to screen those 10 in the ASEA assay. All of these PFAS were less potent than 17-beta estradiol, meaning that their AC50 values were higher than the native ligand. And the important structural features seem to be acrylates and N-alkyl perfluoroalkyl linear sulfonamide structural categories that were significantly associated with this ER activity. Adding in the ASEA assay as another orthogonal assay to confirm specificity indicates very few PFAS with transcription factor and functional ER-dependent cell proliferation. And I've copied the structures of the substances that are highly related that appeared to affect both that transcription factor activity and then the downstream ER-dependent cell proliferation. And finally, PFOA activated ER alpha trans and ER response element cis, but it failed to produce functional estrogen receptor dependent cell proliferation in the ASEA assay. And this was really interesting and points to the fact that we might want to do this kind of confirmation in an ASEA assay before assuming that transcription factor activity might be relevant for downstream responses. And of course, as you might expect, we did observe some PPAR activity for a subset of the PFAS. The trans assay contained endpoints for all 3 human PPARs, alpha, delta, gamma, whereas the cis assay contained the reporter gene controlled by the PPAR response element that responds to all 3 of those PPARs. And again, you can kind of view the PPAR response element as being orthogonal to any one of those PPAR isoforms in the trans assay. The functional groups that were enriched within the active space were mostly carboxylates, along with sulfonates, sulfonamides, and a phenyl ketone, which all have a negative ionic charge at physiological pH, and this is consistent with what's known about ligand binding for the PPARs. And there really wasn't much activity at PPAR delta, maybe because of its smaller binding pocket. You can see here that I've copied figure 6 from the manuscript, and I'm just showing you a little bit of the data for some specific PFAS that were active, and they have differential activity across the PPAR isoforms. So in green is the PPAR response element, and you can see all 4 of these PFAS activated the PPAR response element. But then there was a lot of activity at PPAR alpha and PPAR gamma for all of these substances. And we only saw a number of limited interactions with PPAR delta, this black line. You can see here x-axis is again concentration in log 10 micromolar. So all of this activity seems to be occurring somewhere between, approximately 1 to 10 micromolar at the very lowest, with AC50s typically occurring between 10 and 100 micromolar. Another interesting finding from the transcription factor activity profiling was activation of RXR beta and alpha. PFAS activity for these receptors was found for about 17 PFAS at RXR beta, and 2 of those activated RXR alpha. These PFAS tended to be linear fluorinated carboxylic acids, which meet defined ligand structural requirements for RXR. And they also activated other targets, including PPAR alpha and gamma and stress pathway targets. And here's an example of one of those PFAS, PFNA, which appears to work through RXR specifically. We did a confirmation experiment using an RXR selective antagonist, UVI 3003, which completely blocked PFNA activation of RXR, whereas the PPAR alpha agonist, GW6471, was ineffective. So we did some confirmation here to figure out that this is really truly a RXR ligand in vitro. So after showing you some of the interesting results from estrogen receptor and PPARs and RXR screening, I want to show you a really brief overview of just the xenobiotic nuclear receptor responses across a panel. And this is summarized in figure 3 in the manuscript. You can see here in this heat map, we've got various receptors and their response elements, and then the fold induction indicated by color in the heat map, with red meaning increase in log 2 fold induction for PFAS substances as well as reference substances. And many of the PFAS, it seems, activated PXR and its response element. There was a lot more activity there. The responses were generally modest when you compare them to the potency and efficacy of relative inducers like rifampicin. None of the PFAS were active at CAR or PBRM, its response element, and this is likely due to the assay design. We just have negligible expression of CAR in the HepG2 cells, and it also may work independently of a ligand. Several PFAS actually activated the aryl hydrocarbon receptor, which is a really interesting finding, but the responses were pretty weak for most of the substances, and we don't actually know the in vivo relevance of these findings. After that whirlwind presentation of the overview of the transcription factor activity profiling results and a little bit of the confirmatory work we did in vitro, I want to tell you a little bit about another targeted Tier 2 screening suite we've applied to the PFAS 150, and it's a phenotypic screening panel using human primary cells. This suite consists of actually 12 different assays, and each of these 12 assays include enzyme-linked immunosorbent assay, or ELISA, measures of multiple protein biomarkers associated with immune response and inflammation amongst a number of other phenotypes. The cell types in these cell culture and co-culture models include a wide variety, and I won't tell you about all of them today because they are not all immune relevant. But just to mention a few, we have assays with endothelial cells and peripheral blood mononuclear cells, B cells, bronchial epithelial cells, fibroblasts, coronary artery smooth muscle cells, keratinocytes, macrophages. So you can kind of hear which might be immunorelevant and which might not be. Each assay system is stimulated with a unique set of signals, including cytokines, activators of inflammation, and other growth factors prior to being treated with test chemical. And one of the phenotypes that this panel attempts to detect is immunosuppression. And the reason that we're interested in immunosuppression for PFAS is that PFOA and PFAS are suspected of being immunosuppressive. This relates to a 2016 National Toxicology Program review of PFOA and PFAS that concluded that suppression of antibody responses in animals had some evidence to support it. This review by Jamie DeWitt and colleagues concluded the same. They tried to reconcile some disagreement in the literature among various opinions of the immunosuppression potential, and they also concluded that PFOA and PFAS may be associated with immunosuppression based on the available data. The data itself is a little bit messy. It's a limited data landscape, and across animals and across strains, sex, the endpoints measures, the doses used, there's a lot of variability. And so it's really difficult to come to a final conclusion even on the in vivo data, let alone trying to predict it with in vitro assay models. But that is what we tried to do. We used this biomap system, which is an assay suite that is run by a company called DiscoverX, and it's that suite of primary and primary co-culture models for assessing phenotypes, including some elements of immunosuppression. And I'm showing here just a subset of the 12 assays that we have. These 3 assays seem to have the immune relevant measures in specific immune cells and measures of interleukins that we know to be immune relevant. So it includes this assay system, SAG, which has peripheral blood mononuclear cells and venular endothelial cells. And what's measured here are things like specific PBMC cytotoxicity, as well as IL-8. In this BT system, we again have those PBMCs, but we also have B cells. And so this can be an important disease-relevant screening model for immunity, but also asthma, allergy, oncology. And here we can look at B cell proliferation and PBMC cytotoxicity specifically, as well as secreted IgG. And then finally, this macrophage or MPHG model includes macrophages and venular endothelial cells. And again, here we're measuring IL-8, IL-10, a number of interleukins that could be relevant. And in thinking of, we're trying to screen for in vivo immune responses, what would we be using this as an alternative to? Well, it could be this animal assay, the T-cell dependent antibody response assay, or TDAR, which measures T-cell dependent IgM antibody response and is sometimes modified to measure IgG as well. And so while we have some elements of that in this assay suite, we don't actually have an intact immune system to work with here. So there are limitations. We used reference chemicals, cyclosporin A, azathioprine, methotrexate, and dexamethasone, and the first 3 strongly suppress IgG production in the BT model, whereas dexamethasone affects IL-10 production in the macrophage model. I'll cut to the chase here. One of the questions people had was, well, is PPAR activity going to drive any immunosuppressive response? Is that at all related? And so one of the first things we did was rule that out by looking at some PPAR ligands, prototypical ligands, these agonists, including rosiglitazone, pioglitazone, gemfibrozole, and this oleoyl ethanolamide, shown across all 12 assay systems, not just the 3 that are immune relevant. And they do very little. You can see the y-axis is fold change, and all of their responses really center around 0. And so PPAR agonists have very little activity across this entire suite. And so any PPAR activity from PFAS is unlikely to drive the response. Our next major finding is that PFOA and PFAS failed to provide support for suppression of IgG in the biomap system at the concentration range that we screened. Overall, neither PFOA nor PFAS seem to show activity similar to those reference immunosuppressants that I mentioned, except maybe PFOA at the highest concentration tested, 60 micromolar, where it clustered with dexamethasone, which suppresses IL-10 in one of the co-culture systems, as indicated here. But the human relevance of this finding is somewhat unknown, even though IL-10 is a cytokine that promotes B-cell IgG production. However, several other PFAS did have activities similar to the reference immunosuppressants in some of the cell systems, including suppression of IgG secretion. So I just want to take a moment to show you a little bit of this figure from our forthcoming paper. So you can see here on the x-axis, we have all of the assay systems from the panel, the diversity plus panel versus fold change. And we have all of our reference chemicals and then PFOA. And you can see there's some activity, especially in the SAG and BT systems for the reference immunosuppressants. That's where they pull away from 0. In the BT system specifically, you can see that cyclosporin A has a huge effect, really suppressing production of some of these interleukins that are really important. And you can also see that azithromycin and this green methotrexate, they also affect some interleukin production. And PFOA maybe affects one interleukin in the system, but not nearly like the reference immunosuppressants. In this other cardiac system, you can see that dexamethasone has huge responses, but the reference immunosuppressants don't really. Dexamethasone does act on some specific interleukins here. And then in the macrophage system, this is particularly interesting. We have some effects of Gen-X and PFOA on IL-10, which is more similar to dexamethasone than to cyclosporin A, methotrexate, or azithromycin, which actually affect IgG. There was a single PFAS that at its highest concentration actually did closely associate with cyclosporin A, including suppression of multiple endpoints in that BT system and the SAG system, looking at strong reduction in secreted IgG and the cytokines IL-17A, IL-2, IL-6, and TNF-alpha in the BT assay. And you can see here in the red again is cyclosporin A, and in the black is this one acrylate PFAS at 60 micromolar, and they do actually track pretty closely together in the BT system. You can also see across the diversity plus panel a little bit of a similar fingerprint for cyclosporin A and this one acrylate. You see it again in the SAG system where maybe this one acrylate looks a lot like cyclosporin A and its effects on some key targets. And then this other system, which is maybe more relevant to dermal toxicity, we see some major effects of this particular PFAS. So I want to end with some overall conclusions for these high throughput screening data with the PFAS 150 library. One is that pretty clearly the molecular weight is important for bioactivity. Higher molecular weight tend to be more active in vitro in our current aqueous media cell-based assays that would be sensitive to volatilized substances. The analytical quality control is exceedingly important, as is the specific salt form of the PFAS and its solvent. In general, the PFAS tend to be either equal to or less potent than the median chemical that's been screened in our ToxCast chemical library for these technologies. And then we do see subsets of PFAS that have activity for transcription factor targets, including aryl hydrocarbon receptor, ER, PPAR, PXR, and RXR. But we believe that orthogonal confirmation is really important in order to believe initial screening results. And so that's why we've tried to combine orthogonal readouts. And then finally, our work in progress on screening in a model of immunosuppression really failed to provide support that PFOA or PFAS induced IgG suppression, although we did see a hint of effects on IL-10. And with that, I would like to conclude this really wordy, fast presentation on some of our Tier 2 NAM data and acknowledge some of the critical people involved in this effort. There are a lot of people involved in the PFAS ORD research team, And in particular, I want to thank Grace Padowich and Richard Judson, who co-lead this project, as well as Keith Hauk, who is the lead author on a number of these publications and was the driver of a lot of the bioactivity screening prior to his retirement at the end of 2020. And I also want to thank a number of individuals from the companies that run these assays and serve as scientific experts for us. And then, of course, all of my colleagues in management within the Center for Computational Toxicology and Exposure. So thank you for your time, and I'll take your questions.
Katie, thank you for a nice presentation. We have time for maybe one or two questions, and I just wanted to point out that we can bring the microphone to you. Is there anybody in the Providence audience who has a question? Perhaps we can see if there are any questions on WUHUVA. Let's see. I know there's a lot of questions popping up for Dr. Schlesinger that we'll save to the panel. I'm wondering if there's any questions yet for Dr. Paul Friedman. Okay. So Katie, I have a comment, I guess more than a question, but I guess there is a question in this. I wanted to ask you what your thoughts are about in the data you showed us with decreased IL-6 and TNF-alpha in your immune assays. We actually see similar kind of trends in the serum from mice that are treated with individual PFAS. And that's kind of counterintuitive to some of the notions that PFAS cause inflammation. And so I just wanted to get your thoughts on that, because I've seen other papers also that have shown in cell-based assays there's decreased secretion of TNF-alpha and IL-6. For those of you in the audience, TNF-alpha and IL-6 are two pro-inflammatory cytokines. I don't know if you want to comment on that, Katie.
Yeah, that's a great observation, and I don't really have any sense of it beyond just the empirical observation that seems to be consistent. I don't know what that means when, often we're being validated or referenced by animal data. So when we look at the animal data, from a regulatory context, there are often endpoints that they find more convincing, like in the TDAR IgM response, right? And so I guess my personal inclination is that in the in vitro space, we need to adopt some more pharmacologically relevant models where we look at perhaps a co-challenge of a cell-based system with a vaccine and the PFAS to look at essentially antibody production or reduced antibody production resulting from that co-challenge. And that will be easier for us to evaluate human relevance, because then we can more directly connect that to the human epi data that are being compiled, where they look at response to vaccination. So I don't have a great direct answer to your question, except that when I think about this and what I would love to be able to do, it's creating that more mechanistic link to something that is highly human relevant in the epi studies.
Great, thank you. I'll just field one more question from the WUHUVA from Dr. Birnbaum. Are the ADAgene assays performed with human receptors?
Yeah, hi, Linda. They are performed with human receptors, and the cell line itself, all of the endogenous cis assays, that's all human because it's coming from the HepG2.
Great, thank you. And we'll save some more of the questions for the panel discussion at the end. Okay, so next I would like to welcome Dr. Carrie McDonough. First of all, it's great to have Carrie back. So Carrie happened to be a doctoral student in oceanography program with Dr. Reiner Lohmann. I had the pleasure of serving as one of Carrie's dissertation members. And currently Carrie is an assistant professor at Stony Brook University. She spends a lot of time doing work with high-resolution mass spectrometry and has worked on some really great methods for analyzing PFAS. She studies the fate, transport, and biological effects of anthropogenic organic contaminants in aquatic environments to understand how the molecular structure and chemical properties of pollutants influence their fate in the environment and their biological effects in aquatic organisms and humans. Her recent work focuses on bioaccumulation and transformation of complex PFAS mixtures in marine ecosystems and in occupationally exposed humans, in particular firefighters. And I've had the opportunity to work with Carrie in a bigger working group studying exposure and health effects in firefighters. So with that, she's going to talk to us today about her findings with regard to disposition and metabolism of PFAS mixtures. Welcome, Carrie. Welcome back.
All right, got a timer so I don't go over. All right, thanks everyone. Thank you so much, Angie, for having me today. So today I'm going to be giving kind of a more analytical chemistry perspective to this problem of human PFAS exposure and trying to widen our lens to look at novel PFASs and whether they're relevant to human exposure scenarios. And I wanted to start by just saying, you know, the story of PFASs in our bodies is really a story of analytical chemistry, I think, first and foremost, in that that's our flashlight, right? Like, we can't protect ourselves from things that we can't see. So, you know, starting in the 70s, we had some inkling. Taves in 1968 analyzed his own blood, I guess, as one does sometimes, and found the presence of fluorocarbon molecules. From there, they found, you know, widespread contamination of human tissues with these fluorocompounds. So this is kind of the first hints in the literature publicly available. And now we kind of have some things that are known, right? So this is kind of the tip of the iceberg that Anna referred to. So we know that PFASs are present in everyone. Particularly the perfluoroalkyl acids. We know, looking at the temporal trends, that we're kind of seeing a wave that probably already crested. And we know that highly impacted populations, there are a lot of them all over the world with kind of unique different exposure scenarios with elevated concentrations that are really being seriously impacted by these contaminants. So things have started to, you know, grow from there. We're starting to learn more from kind of the initial mass spec abilities that we had. You know, our analytical chemistry is getting better and better with our, you know, new analysts and fancy machines. And we're finding more and more of these compounds in the environment. And all these different novel PFASs, really a dizzying array. This is just a couple examples. And really the take home is that there are thousands of these things. We know that now. And the question is, how many of these things are in our bodies then? How many of these things are bioaccumulating in humans? How relevant are they for human health? So what we know to date, a lot of it comes from highly exposed communities. Some of the early evidence was from occupational exposures among firefighters. Rotander et al. In 2015 found a bunch of different substituted perfluoroalkyl acids in a small group of Australian firefighters. And more recently, a lot more of these studies are starting to pop up as the analytical capabilities are making that a reality, including this recent one from Kotlars et al. In 2020 of residents in Wilmington, North Carolina, looking at some of those novel ethers, which also, it turns out, are in the blood of these residents. Also alarming is, you know, from the other side, looking at the total organic fluorine question. The evidence there points to there being a significant fraction of this stuff that we still can't identify, and this is, you know, in highly impacted populations as well as the general population. So this is a widespread issue of this kind of dark matter, novel PFAS problem. So I want to make the case that we really need to understand the identities of these novel PFASs to answer a lot of our really pressing questions. The main questions I often hear from people in communities are things like, how are we being exposed and how are we affected by these compounds? And to really answer that, we have to have a comprehensive understanding of the composition of the mixture in their blood. And that can help us, answer those two questions as well as all of the questions in between that we need to answer to really answer those questions about source, fate, exposure, dose, susceptibility, and finally effects. So to do this, we need comprehensive PFAS analysis, which is basically kind of like a unicorn. There's always going to be a trade-off between selectivity and comprehensiveness when you're doing analytical chemistry. And every sample preparation or analytical method choice that you make is going to narrow your window, right? So when you do your sample collection, the choice of matrix, are you doing serum, are you doing whole blood, are you doing tissues, what are you doing? How are you storing it? What's your extraction solvent? What's your extraction method? Are you, selecting just for anions? Are you getting the cations? Are you getting the Zwitter ions? Where are your neutral volatiles going? All of these things are going to impact your ability to really understand someone's exposure and what's in their blood. And finally, of course, your sample analysis. So your chromatography, often you're going to lose your ultra short chains, your ionization choices, and whether or not you have analytical standards. So this figure on your right is showing kind of all the different analytical techniques that we have for these different portions of PFAS and inorganic fluorine, organic fluorine. And today, the approaches that I'll be talking about are really using LC-HRMS, so LC amenable novel PFASs, and targeted. And we're always trying to work on making that little red box bigger by expanding our targeted methods, and then also combining that with high res to kind of be more comprehensive. So I'm going to start by talking about a human biomonitoring project that I was a part of called PFAS Aware. This is a community in El Paso County, Colorado, south of Colorado Springs and the Peterson Air Force Base, where PFAS and PFOA exceeding the HAL were found in drinking water in these communities between 2013 and 2016. We know high levels of exposure ended in 2015. We don't yet know exactly when they began. So this is a study of analysis of water and residential exposure in these communities. It was a collaboration with Colorado School of Public Health, John Adgate and Ann Starling, as well as Chris Higgins at Mines. And my part of this study was really to develop methods for comprehensive characterization of the exposure source and the human internal dose, the serum, in this AFFF impacted community. So this is a community where we know they were impacted by an aqueous film forming foam type emission getting into the drinking water. So I'm going to start by just showing the perfluoroalkyl acids here. So this is a targeted analysis, kind of the first things that we can learn. And the blue and orange bars that you see, that's drinking water. So this is raw drinking water from public and private wells, going from north to south, from up to down in these different water districts. So you can immediately see the source zone was to the north. And there's a pretty clear gradient showing that. And this really highlights that these different communities are going to have very different exposure levels depending on which water district they're in. And that's reflected pretty clearly in their mean serum PFAS concentrations, which are in the red. So we've got the total carboxylates, the hexane sulfonate, and the octane sulfonate. And some takeaways for this, I mean, clearly concentrations of these PFAAs are varying among water districts, and they're much more elevated in the close areas to the source zone. And also we have very highly elevated hexane sulfonate concentrations, 12x NHANES. So this is a kind of unique cohort in that regard. So I want to move on to really focus on kind of the more novel compounds and what we've learned about them. So doing suspect screening on these human serum samples, I found a peak that was detected in about more than 80% of our human serum samples. We've got about 200 participants in the study at MZ 460.9. That was matching to one of our analytical standards for PFEX, which is the cyclic C8 with the little C2 tail and the 6-member ring. And that compound actually was recently found in a study of Swedish serum, Miaz et al. 2020. So we thought kind of maybe that's what it is. But when you look at the fragmentation, it just does not match. So fragmentation for PFAX pretty cleanly has these two major signature fragments. And when we looked at our fragmentation, we see kind of a more complex picture. There seem to be a lot of low-level fragments indicative of perfluoroalkyl chains. So I ran a more concentrated pooled serum sample to target that mystery peak. And even though with a lot of PFAS this is really challenging, you just get really low fragmentation even when you use a high collision energy. I was able to assign a lot of these peaks pretty good annotations and pretty confidently, tentatively ID this as a unsaturated PFAS. So we removed 2 fluorines at a double bond, something like that. This could represent multiple isomers or branching, not entirely sure on the positional isomers here. As far as I know, it has not been previously identified in human serum, and semi-quantitative concentration estimates put it at about 0.03 to 2 nanograms per mil, mean of 0.3. So widespread, low concentration, and correlating pretty well with the other known AFFF-associated PFASs in this community, like PFOS and hexane sulfonate, so likely a similar source. UPFOS was also found in the drinking water. So here's the UPFOS semi-quantitative estimates for all of the drinking water, going again from south to north. And you can see that same gradient going towards the source zone, more or less. And we can use those drinking water concentrations to then calculate serum water enrichment ratios and kind of look at the distribution of, how these residents, what their serum looks like versus their drinking water. So this is for Security and Wide Field, which are the kind of the northern region near the source zone in this area. And we found that UPFOS was accumulating in blood to a similar degree to the other C8s. So it seems like maybe some kind of chain length dependence, and you can see the short chains tend to have lower enrichment, which we know. I also wanted to point out we found additional novel substituted perfluoroalkyl acids, keto and oxygen substituted compounds in the serum, and those were not detected in the water at all. Another thing we can do, now that we have semi-quantitative estimates, is compare trends over time. So we did take a second subset of blood samples from just a subset of the residents in June 2019. So this is for 50 residents. And there were clear declines in a number of compounds, pretty small number of people, so we can't really do any kind of half-lives or anything, but you can see that there's a decline, less so for the short chain C5, perfluoropentane sulfonate. And the trends are less clear for unsaturated PFOS as well. So we have some that are kind of increasing and some decreasing, but overall, looks like a decrease after the problem was mitigated and the water was clean. So kind of switch gears here. I kind of wanted to also point out these substituted perfluoroalkyl acids. We've also found them in other cohorts. So this is the work that Angie was referring to. I've been collaborating with the Burgess group, Jeff Burgess at University of Arizona, to also look at firefighters. And the kind of overarching goal of this is to look at how their serum concentrations respond to fire events. But I'm not going to get into that today. I'm just going to kind of show you a little bit of the major novel PFAAs. So we developed a new serum analysis method to work with these firefighter blood samples, trying to get something more comprehensive. It's a lipid removal method, high throughput lipid removal method. This is my student, Jen, who's been working on this. Pretty happy with the method. So far, we validated for 40 PFASs. Growing analyte list. Our spike recoveries have been pretty nice, and just trying to expand, what we can regularly monitor. So along with that targeted list, we also did suspect screening on these samples. And here we're seeing one of these, again, substituted perfluoroalkyl acids as one of these, 171 peaks that we found in our suspect screening. So this is the keto-substituted perfluorooctane sulfonate, tentatively ID'd in more than 90% of our firefighters. It was a small cohort, about 20 people, so not a lot of data. And the semi-quant suggests really low levels, again, like 0.2 nanograms per mil. You can see it's in the baseline and the post-fire, so no differences with their activity, but it's just there. So we're seeing these widespread, low-concentration substituted compounds. So now I want to delve into what we can learn about those using other models. So what can we learn about our human biomonitoring data looking at a mouse model that was dosed with AFFF? So this is work I did in collaboration with Jamie DeWitt at East Carolina University. And she conducted a study where she dosed mice for 10 days with an AFFF formula. This is a commercial product, not really what someone would drink, but representative of the commercial product, complex mixture. And we collected serum. After a 6-day deprivation, we also collected urine throughout the study. So here's the bubble plot of kind of all of the different novel PFASs that were found in the serum after deprivation. So all the different colors are different homologous series. You can see the kind of nice diagonal line that kind of represents a PFAS homologous series. And the size of the circle represents kind of the relative peak area. So it's some indication of the abundance of the compound. And I've got all the structures along the side there, so you can see everything that we kind of found that was significant here is a substituted perfluoroalkyl sulfonate. So we've got cyclic unsaturated, hydrogen substituted, oxygen substituted. And the main takeaway of why I'm showing this is that 3 of these compound classes are what was detected in serum from our residents of El Paso County. The keto was also in our occupationally exposed firefighters. And there's the ones that we found in El Paso County specifically, UPFOS, OPF-HPS, and KPFOS. You can see 2 of those are extremely prominent. Also, they look like they're some of the things that were more highly enriched in our mouse blood. So now if we look at relative enrichment, what we're looking at here, we've normalized everything to PFOS. So if something's near that little dashed line, near 1, that means it's kind of enriched in blood to a similar level as PFOS in our mouse model. If it's higher, that means it's enriched to a higher degree than PFOS is. And what's interesting here is we've got a number of substituted compounds that kind of look like they're enriched to a similar extent. And then we also have KPFOS and UPFOS, which we're becoming familiar with now, at these much higher levels, which to me indicates either these compounds are more bioaccumulative than PFOS, or they are forming in the body due to the transformation of some unknown precursors. I also wanted to add the OPF-HPS didn't even have a peak in our AFFF for a BAF calculation, so also seemingly enriched in the mouse blood. So to get at this question a little better, the next thing we're trying is looking at novel PFAA formation during metabolism. So this is kind of an initial query that we're just starting out now to look at whether these substituted PFAAs that we're seeing enriched in the mouse serum could form due to metabolism in the liver. So what we're doing here, this is work that my PhD student David is doing, and also big thanks to Allie Phillips and James McCord who helped us with the protocols. We had never done S9 assays before. So we're taking 5,000x diluted AFFF, so complex mixture, incubating it with these S9s, quenching at time 0, time 60, and then just running it by QTOF mass spec to look for novel compounds. So it's really just a first kind of brute force method to look at, do these things form? So doing this, we have a number of compounds that don't change, which isn't surprising. So PFOS, no change. That's actually quite comforting because we use it as a benchmarking compound a lot in these AFFF studies, so knowing that it's not changing is good. UPFOS is a little inconclusive. It looks like there's a number of isomers present, so it's a little difficult to say. Probably going to need to get better separation on that one. ChloroPFOS is another one that, if you remember, the bioaccumulation factors came out right around 1. Similar bioaccumulation, similar enrichment to PFOS. And here we're also seeing, doesn't look like it's being formed or anything funky is going on, at least in the liver with liver incubations. So what's interesting is what we're seeing that looks like it's really unequivocally being formed here. So perfluorohexane sulfonate, we know there are a lot of hexane sulfonate precursors in these AFFFs. Really sizable increase in the peaks here. So the red is post-metabolism. The black, which is kind of dark red here, is before, so the time 0. And we also have a number of these substituted compounds coming out. So the keto, the oxygen substituted, and a hydrogen substituted that wasn't even there when we started. Additionally, we have a lot of 6,2-FTS. This is a field-collected AFFF. It's a little bit of a mystery, but appears to have a significant amount of fluorotelomeres in it as well. And Yeah, preliminary. These are pretty new data. Still kind of puzzling through them, and I'd love to talk more to everyone about them. So from here, I mean, the next question I had was, where is this stuff coming from then? Where, there have to be precursors that are transforming in our assays and in our mice, and perhaps in our own bodies, that could be forming these things. So that's where we get to the PFAS precursors. I put the slide in because I wasn't sure how much they'd be covered. I think everyone's pretty familiar. I call them pre-PFAAs, just to make it more specific, because we're talking about compounds that specifically go to PFAAs. Labelability can be different in different environments, which is good to keep in mind, and really good to keep in mind that some products of these precursors may not be included in routine methods. If these things are forming, they wouldn't be caught in a targeted method. Are they relevant? Hard to say. We know our forever chemicals are human relevant. There's a number of widespread pre-PFAAs that we know are relevant. These more complex precursors that might be present in something like an AFFF that we're feeding to a mouse in a mouse model study, do they actually make it to a point where they're being ingested? I think this is a really good question. They're also in a lot of our consumer products. So in that case, they're kind of right on your skin or maybe even in your mouth, and they probably are relevant. We know that there are a lot of these compounds in our AFFFs. So this is a study I did with Jeremy Komel, where he used a fluoromatch to look for a bunch of different compounds in our AFFF. And you can see there's a lot of different novel PFASs, and a lot of them are precursors. So we know there are precursors. We know a lot of them are C6 precursors. That part is kind of known. And we also know that there are pre-PFAAs in drinking water. So this is back to our El Paso County cohort. And we're looking again at this gradient in drinking water of public and private wells throughout this area, north to south. And again, you see the gradient. And now we're looking at our perfluoroalkyl sulfonamides. So these are precursors to the sulfonates. And they're clearly there. The C3 to C6 are the most prominent. There's a really steep gradient here. The C6 in particular was only detected in security. So it looks like it's really highlighting that not only is the concentration different, the exposure concentration among these different districts, but also the mixture. And then when you think about how complicated mixture toxicity of PFAS is, it's really, it really gets to a complex problem of how this is affecting residents who are drinking this water. So what about in human serum then from El Paso County? So what you're looking at here is bar charts of the composition of year 1 and year 2 serum for our carboxylates and for our sulfonates, and then also drinking water. And the little hatched marks represent the summed precursors that we found in the drinking water for each of these chain lengths. And it's pretty clear right away, they make up a pretty small fraction of our drinking water, and they are not found in our human serum. So metabolism, excretion, transformation prior to ingestion, during advanced drinking water treatment processes, things like that, all of these are possible. What we do know from mouse studies, though, is that it seems pretty clear these things can transform in the body. So what you're looking at here is an AFFF spiked into a mouse serum, so no metabolism of any kind. In the bubble plot there with the big gray bubbles, those are all sulfonamide precursors. And then we see our post-deparation mouse serum again, they're all gone. So what that tells us is some of these perturbations we're seeing in the bioaccumulation profiles are likely caused by in vivo transformation of these precursors. Though, we could also have rapid excretion to some extent. So urine is important to monitor in these types of studies. So another, piece of evidence in this puzzle is when you look at the relative enrichment of known PFAAs in mouse serum. So this is the same AFFF dosed mice. And now we're just looking at the perfluoroalkyl acids. And we know usually there's a chain length dependence when you look at bioaccumulation factors for perfluoroalkyl acids, for perfluoroalkyl sulfonates specifically. But here we're seeing this really enhanced enrichment of PFHXS and also HPS, which is a bit puzzling. But likely these are products of pre-PFAA biotransformation in the body in the mouse model, and that reflects, agrees well with our in vitro assay using the same exact AFFF. So hexane sulfonate formation is very likely here. So what else is puzzling, though, is that the pre-PFAAs that you might expect, the usual suspects in our suspect screening, don't seem to really be the culprits for what we're observing, at least in our in vitro assays. So, we looked at a number of the compounds that we had seen in our AFFF that were not present in mouse blood, and most of them were remaining unchanged in our in vitro assays. So what this points to me, is that we have further need for a really deep dive into non-target to try to figure out what's going on here, if there are novel precursors that we actually haven't identified yet, which seems pretty wild considering how much effort has been put into the non-target analysis on these AFFFs. And we also have to mention that those substituted compounds I was showing as products wouldn't form from these anyways. PFHXS would. A lot of those other products, we don't know what compounds, we don't really have compounds that would form those. So lots of question marks. So what have we learned? I think I've at least demonstrated at this point that to really understand PFASs in human serum, particularly in these highly impacted communities, we need high-res mass spec. We need to be using high-res mass spec all the time to get at that novel PFAS question. And we need to keep developing better methods to be more comprehensive. We also have to combine what we're observing with in vivo models and in vitro models using complex mixtures, environmentally relevant, exposure-relevant mixtures, to really get at these questions, because there aren't neat standards for these compounds. It's the only way you can study them. We saw that members of impacted communities are exposed to complex, temporally and spatially variable mixtures, which is a huge challenge when you look at epidemiology. And the substituted PFAAs appear to be widespread, bioaccumulative. They might even form in vivo, but they do not make up a large portion of the unexplained organofluorine. That we haven't answered. They're really low concentrations. That can't be the explanation. And also this work highlights that there's much to learn still about the precursors, or the PFAAs, pre-PFAAs, and their relevance for human exposure, their toxicokinetics, and their effects in humans. So, at this point, it's kind of raising more questions maybe than it's answering, but I think we're really uncovering some of the directions that we need to go in, and that's progress. So, the next places I really want to go are, what are the major sources of exposure to novel PFASs, especially pre-PFAAs? How do the toxicokinetics and bioaccumulation potential of these novel PFASs compare to well-studied structures? What's the impact of transformable pre-PFAAs, these precursors, on internal dose and bioaccumulation profiles? How's that perturbing what we're seeing when we're in the field? And are these pre-PFAAs and other novel PFASs significant for human health risk assessment? Do we have to take them into account? Probably. So with that, I want to thank all of my collaborators without whom this would not be possible, as well as my wonderful research group at Stony Brook University and my funding sources. And I can take questions. Thank you. Okay. So we can do it like naturally now. Yeah.
Thank you, Kerry. This was a great talk. I was just wondering, when you were discussing the questions about the precursors, the PFAA, and you were comparing the AFFF concentration of the precursors and with the final degradation product, did you also do any type of total fluorine analysis on those samples to have some mass balance, to know if you are gathering everything what was spiked to the, or what did you feed to the mice? I'm not sure if my question was clear.
No, I think I gotcha. I mean, when I look at the in vitro data, the first thing I think is the next thing we need to do is work on the mass balance and try to do, I think the next thing we're going to do is top assay just to see kind of what that looks like. on that AFFF and try to get as good of a characterization of the AFFF as we can. Looking at the numbers, I would say, I mean, they're high. Like the hexane sulfonate formation is high, but some of those AFFFs, I mean, 50% is precursors. If most of that is C6, like it's not unbelievable that you would form that much hexane sulfonate. So it's not too crazy, but yes, mass balance is key as a next step. So yeah, probably top assay coupled with high-res mass spec to try to get at some of those questions.
Thank you.
Great talk and very interesting work. This is not on. There we go. Great talk and interesting work. So a question, I may have missed it, but did you propose any structures, any chemical structures for your unknowns? And then part 2 question is, did you use any workflows, or did you just, after the blank subtraction, you eyeballed and found those peaks and then just interpreted the mass spectra?
That is a great question. So on the proposed structures, we have tried. So when we saw the mouse data for the first time, we basically just went and made theoretical structures for precursors that could form those substituted compounds and made a suspect screening list and tried to screen for them. Nothing definitive from doing that. Besides that, yeah, it's an ongoing question. I think, yeah, more non-target might be the way to go there. For the workflows, it's evolving over time as I'm, learning more and more about non-target workflows. The UPFOS work was pretty much just, we saw a peak of interest and kind of put our nose to the grindstone and worked on it for a really long time. Usually with suspect screening, it's a little less needed to have like a real high-res mass spec workflow because you can use your suspect screening list and you get your library matches. But we are also doing the FLUORO match now, which I like the agreement that we've gotten between manual annotation and FLUORO match. So that's one direction. And yeah, exploring some of the other workflows as well. Absolutely. Yeah, we need GC methods. It's becoming very clear that we need GC methods to look at a lot of this stuff. Yeah. Thank you.